Thank you. Thanks so much for inviting me. I'm actually a native New Yorker. I was born on the Upper West Side, and I grew up in Westchester, so I've been in two hotspots for the virus, but been in Philadelphia for quite a while now. So coronaviruses are really adept at antagonizing the innate immune response, and I'm going to be talking about that. But first, I'm going to give a really short history of coronaviruses, coronavirus research. Then I want to talk about some of the many proteins that coronaviruses encode that, that have really just very many anti-host evasion or antagonist activities. And then I'm going to talk about like antagonism of the host interferon response, giving examples both from mouse hepatitis virus and from MERS, the two viruses that we've worked with up until the recent epidemic, and that we've recently added the SARS-CoV-2 to our repertoire. Here's the timeline of coronavirus research, or coronavirus dating back pretty much from when I started my lab in 1980. So at the time I started my lab, we knew that there were human coronaviruses that caused the common cold, and we knew that there were very many animal coronaviruses infecting just about every species of animal, and there was a lot of vaccine development done. I can find papers back to the 1930s on infectious bronchitis virus. So I call this whole era from the late 60s and 70s, the studies of animal coronaviruses, vaccine development, human cold viruses, and a lot of work was done on the mouse hepatitis virus model. So back here, we'll just say we knew about OC43 and 229E, the now known common cold viruses. OC43 can also occasionally infect the lower respiratory tract. And during this period between then and 2002, when SARS emerged from southern China, there was a lot of work developed on understanding how coronaviruses entered cells, their very interesting and complicated method of expressing their RNAs and proteins, and really what these very many proteins were that I'll talk about in a minute. But things were pretty quiet during that period. There weren't a whole lot of people working on these viruses. Much of the work was done on MHV, most of the molecular biology, and also some on IBV, infectious bronchitis virus, and bovine coronavirus. So after we realized that SARS probably originated in bats and was transmitted probably to civets and then to humans, there was a lot of research done, well, I'll call this the pathogenic human coronavirus period. And people looked for other coronavirus, other human viruses, and found HKU1 and NL63. And I should mention here too, and these viruses were more pathogenic than cold viruses, but not lethal in the way that SARS was. And I should mention that SARS only lasted for about 7 months. By the July of 2003, it was pretty much gone. But there was a lot of looking for other viruses at that time. And the other important thing that happened was that people started searching for more coronaviruses in bats. And as we know now, there are just many SARS-like viruses found in bats, and other coronaviruses as well. And then things, I mean, at this point, many people rushed in to work on coronaviruses, many HIV researchers, flu researchers. And then things were pretty quiet until 2012, when MERS emerged in the Middle East and Saudi Arabia, causing another epidemic. MERS, of course, came from bats, has a reservoir in camels, so different from civets, who we think were sort of a transitory host for coronaviruses. MERS coronaviruses are found in camels, both in the Middle East and in Africa, and they continue to be transmitted into humans at a pretty slow rate. But the MERS epidemic is still going on. So then after MERS, things were quiet again in coronavirus world until, as we all know, in late 2019, SARS-CoV-2 emerged from in China again. So we now have 3 severe respiratory diseases caused by coronaviruses, which was, in retrospect, was quite shocking to the early coronavirus community that was used to these cold viruses and these model coronaviruses. But by now, we know that coronaviruses can cause this obviously severe disease, which we call this COVID-19. The other just sort of interesting fact was that the first coronavirus meeting was held in Würzburg, Germany in 1980. There were 60 people there. I went back and looked at the book. the next meeting I can mention is and then we would have these meetings every few years. The 10th meeting was in the Netherlands in 2003. And this was just after SARS, and this attracted a huge number of people. And there was supposed to be the 15th meeting in the same place in the Netherlands, which is kind of eerie, in 2020, but this meeting was postponed due to the epidemic. Okay, so just let me get into the innate immune antagonism. So this is just to show that this is an example from MERS, that during infection, this is in A549 cells, there's very little induction of interferons. Here's interferon beta and lambda. It's after 36 hours, just 5-fold or 10-fold induced. And just in contrast, here's the same kind of experiment with Sendai or Synbis, two other RNA viruses in different classes of different families of virus. Interferon beta is induced like, this is a log scale, this is a linear scale, about 1,000-fold, interferon lambda, almost 100,000-fold, and OAS-2, about 100,000-fold. So MERS and other coronaviruses are really, really good at restricting interferon-like responses. So I just want to, the next couple slides, I want to talk a little bit about the coronavirus genomes and the proteins that they encode. So this shows the genomes from the coronavirus families or groups or lineages that have human viruses. The alpha coronaviruses include 2290 and NL63. The beta coronaviruses there are 3 lineages, A, B, C. They now have these new names, but it's much easier to think of A, B, C. The lineage A viruses include OC43, HKU1, and MHV, the mouse virus. The B lineage is SARS-like viruses, and the C lineage is MERS-like viruses. So all these viruses have very similar genome structures. They all have this 20 kilobases, 2/3 of the genome devoted to replicase-associated proteins, which I'll talk about more. There are 16 of them, non-structural proteins. And then the spike protein and the other structural proteins that you've heard about today are all arrayed in the same order, E, M, and N at the 3' portion of the genome. Pretty soon, right after SARS was identified, SARS-2, it was quickly identified to be very closely related in sequence to SARS-1 and have the similar gene arrangement at the 3' end of the genome. These 3' genes interspersed among the spike protein or the structural proteins are the so-called accessory proteins, and they encode many of the interferon antagonists. And each one of these are different for each lineage of virus, and I think of them as sort of giving each virus its kind of characteristic difference from the other ones, because really most of the genome is well conserved. The genes are the types of genes. There are obviously mutations and differences, but these genes are very closely related, except for these accessory proteins, which are completely different among the lineages, as you can see here. So I'm going to talk about, let me just talk a little bit about the proteins encoded in ORF1A and ORF1B, these conserved replicase proteins. We all know about the RDRP, the polymerase. There are 2 proteases encoded in ORF1A that process the polyprotein as it's being translated. But there are all these other proteins, and these proteins are also kind of part of the replicase complex in replicating RNA. But then we have all these proteins here that have other functions in antagonizing the host. There's the XON we've heard about, the proofreading enzyme. This is incorrect. This should be over here. There's Endo U, which I'm going to talk about later, NSP15, which prevents the synthesis of accumulation of double-stranded RNA and host responses. There are 3 capping proteins here, and here, the NTPase, and these are all to sort of shield the viral RNAs from recognition by the host, antiviral activities. And then there are also multiple host antagonist activities encoded in NSP1 and in NSP3, a deubiquitinase and a macro domain. So all of these activities in blue are really directed towards promoting synthesis and stability of viral RNAs, capping of 5' ends, and protection from host cell sensors and interferon response. So all coronaviruses encode these same activities. I'm going to talk about some of the accessory proteins encoded by MHV and MERS coronavirus. So these are lineage A and lineage C, not different from SARS. These are from previous works in my lab. We're going to talk about NS2 and NS4A and NS4B. These are, again, specific to these lineages. And then I'm going to talk about NSP15 as well, the Endo U protein. And in some cases, these proteins have redundant activities, both the accessory proteins and the conserved replicase-associated proteins. So now I just want to review the double-stranded activated antiviral pathways that these proteins interact with and antagonize. So early in infection, coronaviruses synthesize double-stranded RNA, as do other RNA viruses and as do some DNA viruses. And this double-stranded RNA is recognized by host sensors. In the case of coronaviruses, MDA5, which signals through MAVs to produce interferons type 1 and type 3, which then induce interferon-stimulated genes which have antiviral activities. But there are two other parallel pathways that we work with in our lab. The OAS-RNase L pathway is something that we focused on for a long time now. So there are several OAS proteins that recognize double-stranded RNA that they then synthesize 2,5A, a small oligoadenylate, and 2,5A, and I'm sorry, should go back here. 2,5A causes the dimerization and activation of RNase L, which then cleaves single and single-stranded both host and viral RNA, and this results in protein synthesis inhibition, apoptosis, and antiviral activities as the viral RNA is degraded as well as the host RNA. The other pathway is PKR. So PKR recognizes double-stranded RNA, autophosphorylates, then phosphorylates EIF2 alpha, a protein synthesis initiation factor, which then causes protein synthesis to be inhibited and also can lead to apoptosis. And so both these two pathways can be activated independently of interferon, but they're also upregulated by interferon because OAS and PKR are interferon-stimulated genes. So the first thing I'm going to talk about is a group of proteins called phosphodiesterases encoded by some coronaviruses that cleave 2,5A and inhibit this pathway. And so I'm going to go on to describe that in some detail. Before I go on, I was just going to mention that this pathway, actually the OAS pathway, we've recently characterized it in bat cells, and it's actually quite similar to human cells. We thought there might be differences, but there weren't. Okay, so coronavirus phosphodiesterases. These are members of the 2H phosphoesterase family. They have two catalytic motifs that are histidine X, serine threonine X. They're encoded both in the beta coronavirus lineage A group of viruses. That includes OC43 human virus. We've mostly worked on this with MHV. And then the lineage C viruses encode NS4B, which actually has some homology to NS2. These are the only two accessory proteins with homology among the different lineages of proteins. But there are some differences, which I'll talk about later. And then toroviruses that are related to coronaviruses also encode a homologous protein at the end of ORF1A. It's called PP1A C-terminal domain. So these are homologous proteins encoded in different parts of the genomes of these three groups of related viruses. So NS2 of MHV, this is a small protein, 30 kilodalton protein expressed in the cytoplasm. It's not essential for replication in vitro, nor is it for central nervous system pathogenesis, which I'll talk about a little bit later. So these are the two catalytic histidines. We early on obtained a mutant virus from Stuart Siddell in Britain. So we compared wild type and mutant in this. So this mutation, it activates the phosphodiesterase activity. So we compared replication of these viruses in bone marrow derived macrophages from mice, and we can see that the wild type virus replicates really well to 6 logs, whereas the mutant virus pretty much can barely replicate at all. However, when we put these viruses into bone marrow derived macrophages derived from RNA cell knockout mice, we see that the mutant virus recovers replication very nicely. we then wanted to see where along this pathway did this inhibition or this rescue of virus occur. So we looked first at the level of 2,5A in infected cells, and we found that in these B6 macrophages that the wild-type virus induced very little 2,5A, whereas the mutant virus induced quite a lot more. So it looked like this would be consistent with its phosphodiesterase activity. We also looked at the actual RNase activity by degradation of ribosomal RNA in a so-called chip assay on a bioanalyzer. So this is kind of a pseudo gel coming off a bioanalyzer. And we can see that in cells infected by wild-type virus, the ribosomal RNA 28 and 18S looks largely intact, whereas in the mutant infected cells, we can see quite a lot of degradation of RNA. We did the same. We also looked at these mutants in mice in our mouse model for hepatitis. So we infect the mice intrahepatically. The virus replicates over the first week or so post-infection, and that gets cleared by the adaptive immune system. But the innate immune system is really important during these early stages of infection. So if we look at replication of virus in the liver at day 5 post-infection shown here, that's the peak of replication. We see again that virus replicates to almost 7 logs in the livers of wild-type virus, whereas the mutant virus can barely replicate at all, shown here. Whereas if we replicate or infect RNA cell knockout mice, we find that the mutant virus can recover completely its replication, as shown here. And this is also reflected in staining of liver tissue from these mice, that the wild-type virus can express antigen quite robustly in livers from wild-type or mutant mice, whereas the mutant virus barely shows any protein expression in the liver of wild-type mouse, but recovers in RNA cell knockout mice. This is nucleocapsid protein staining. And the same thing is true if we look at H&E staining. There's really no pathology in the mutant virus infected wild-type mice, whereas we see quite a lot in the RNA cell knockout mouse, and the wild-type virus can produce similar pathology in either mouse genotype. And as I mentioned before, interestingly, if we do the same experiment, except doing an intracranial infection, the brain is the other major site of infection of MHV, we can see that replication really doesn't depend on the PDE at all. So the mutant virus replicates similarly to wild type in B6 or RNA cell knockout mouse. And here you can see just in the same mouse, the virus is restricted in the liver of B6 mice, but not RNA cell knockout mice. And so from the system, we learned quite a lot about activation of RNA cell. And one of the things we learned, I don't really have much time to go into it, obviously, but one thing that we did learn was that we looked at the levels of OAS protein expression in the brain and the liver, and we could see that the expression was much higher in the liver than in the brain. RNA cell is constitutive, it's not an ISG. So and we found by looking at a lot of different cell types that activation of this pathway really depended on basal levels of expression of these OAS molecules. We also found that in general, the brain has really low expression of interferon stimulated genes compared to the liver. This is in uninfected tissue. So the activation of these pathways really, it doesn't really matter that virus can induce higher levels of these OASs. It all depends on what's true at a basal level. So now MHV Endo U or NSP15 also antagonizes RNase L, and I want to discuss that just for a little bit. So we were able to obtain a mutant. This was work we did with Volker Thiel in Switzerland. He had made a mutant of Endo U, of NSP15, and we compared the replication of that mutant and wild type again in macrophages. This is data on a paper with Volker, and we can see that again, very similar pattern, that the Endo U mutant doesn't replicate very well at all, while the wild type mutant replicates quite well. So both Volker's lab and Susan Baker's lab showed that the Endo U mutants produced a lot more double-stranded RNA in infected cells. So And it wasn't really known what the substrate for Endo U was. So here we infected bone marrow derived macrophages more recently, and we stained both for single-stranded RNA by FISH and for double-stranded RNA using an antibody. And we can see that as Susan and Volker had published, that there is more double-stranded DNA in the Endo U mutant infected cells, but there's quite a lot more of single-stranded RNA. So even though the virus replicates quite poorly, it's actually producing a lot more single-stranded RNA. And in a recent study in collaboration with Volker and with David Barton, this is under review at the moment, we found that that Endo U actually cleaves its own, it cleaves viral RNA, and that would be the outcome of that would be the cleavage of single-stranded genome RNA, and it was only plus-strand genome RNA that was cleaved, would result in less accumulation of double-stranded RNA and less activation of the pathways. And so here, this just shows, similar to the NS2 mutant, that the Endo U mutant also activates RNA cell that's shown here. Here's the wild type virus, and here's the mutant virus infected cells. You can see activation and degradation of ribosomal RNA. But differently from the NS2 mutant, these mutants don't recover in RNA cell knockout cells, but they do recover in RNA cell PKR double knockout cells. So Endo U and NS2 kind of overlap in their effects. Well, just to summarize this section, double-stranded RNA is recognized by OAS to activate RNA cell, but it can be restricted right at this level here. So if we knock out Endo U, we get activation of the pathway. OAS produces 2,5-A. This is another step at which MHV can knock down the pathway, which prevents RNA cell dimerization, activation, and we get replication and pathogenesis. So these are two different ways that, that the virus can inhibit this pathway, and either knocking out either one of them activates the pathway. So this suggests that both of this pathway, at least in the mouse, is really important to restrict MHV in the mouse liver. And it's taking a lot of, it's actually just this protein, the NS2 protein's only function that we know of is to knock down this pathway, whereas Endo U has, is a more general function of anti-interferon function. Now I want to move a little bit to MERS. So we have MERS encodes NS4A and NS4B over here in the genome. And NS4B, as I mentioned before, is a phosphodiesterase with homology to NS2. But NS4A is a double-stranded RNA binding protein, which would be predicted also to restrict the activation of the three double-stranded RNA-induced pathways. So just a little bit about these PDEs. So here's NS2. It's expressed in the cytoplasm, shown here. But MERS NS4B has a nuclear localization signal on its amino terminus, and it is expressed mostly in the nucleus, but some in the cytoplasm. So this was of interest to us because we'd shown previously with ACAP7, which is a cellular phosphodiesterase that was expressed in the nucleus, that this was actually unable to inhibit RNA cell. And we thought that that the protein would really have to be expressed in the cytoplasm to work as an RNA cell inhibitor and to help the virus to evade the RNA cell pathway. So we wanted to know whether this NS4B protein was actually an active RNA cell antagonist. So we took our reverse genetic system for MHV with a backbone with a mutant NS2, so an inactive phosphodiesterase. We replaced NS4B, which is a not essential protein in MHV. This is all MHV. We expressed either wild type MERS NS4B or mutant MERS NS4B, shown here, and within the OR4, and then we compared replication in both wild type and RNA cell knockout macrophages. So these are just controls that you saw before. This is MHV and mutant MHV that recovers in RNA cell knockout cells. And these are the chimeric viruses. So this virus expresses, this has an NS2 mutation expressing MERS NSb, and it replicates quite well in wild-type macrophages, whereas the virus expressing mutant NSb does not. And both viruses recover in the RNA cell knockout mutants. So mice, and here we can see on a chip assay that the MERS NSb wild type 4b protein protects the cell from activation of RNA cell, whereas the mutant MERS protein does not. So, so clearly this NS4B protein, although expressed primarily in the nucleus, is able to antagonize RNA cell and rescue virus replication. We had similar data. We could actually express all the other lineage A beta coronavirus NS2s, for example, OC43. We could also express rotavirus VP3, which is also a phosphodiesterase, and host ACAP 7 phosphodiesterase within this backbone of mutant MHV, and each one of them could rescue replication. So all of these function as a phosphodiesterase and RNA cell antagonist within the context of MHV, both in vitro and also in vivo in the mouse liver.
Couple minutes, Susan.
Okay, I'm almost done. So mutation of, let's see. Oh, okay, I'm not almost done. Okay, let me quickly, I'll just show this then. We also made mutants of NS4B in the virus itself, and that's shown here. We had an NLS mutant and a phosphodiesterase mutant. And we can see here, we looked at whether mutating NS4B in the virus particle would promote interferon, would promote RNA cell activation. And we see here that there's a very small amount of activation in the phosphodiesterase mutant, nothing in the NLS mutant as expected. And finally, that we also looked at the NS4A and NS4AB mutants, NS4A being a double-strand RNA antagonist. So we saw very little activation of this pathway, very different from what we saw in MHV. We also saw with a delta 4A, NS4A double-strand RNA binding mutant, also very little, we saw a small activation of PKR, but nothing like our SynBis virus control shown here. So I think I will just skip that if I'm right near the end. Well, just to say that these NS4AB mutants, MERS mutants, still activate interferons only very weakly. We're talking about interferon beta to 4-fold versus what we saw earlier with the other viruses. For mutants, activate interferon lambda somewhat better, but still very weakly compared to SynBis virus shown here. So I will just, there's also, MERS also encodes an Endo U that we said before cleaves genome RNA, resulting in less double-stranded RNA. So just summarize real quickly, the Endo Mu mutant also induces very little interferons and doesn't even activate PKR. So the conclusion from this is that MERS encodes several antagonists, and single mutation of any one of them really has very small effect. So they likely work together to really knock down these pathways. Just to conclude here, here's the pathways again. We know that NS2 and NS4B act at this step, preventing RNA cell activation. NS4A binds to double-stranded RNA and has some effect on knocking down these pathways. And Endo U also causes a reduction in the accumulation of double-stranded RNA. But these proteins have different effects in the two different viruses. So just to summarize, coronaviruses encode multiple interferon antagonists in conserved non-structural proteins and in lineage-specific accessory proteins. In the MHV model, both NS2 and Endo U are both required to prevent activation of this one pathway. Both are required for replication in the liver, but it's an organ-specific effect, and they're not required in the brain. MERS coronavirus expresses two accessory proteins that, when deleted, result in weak activation of all three pathways. Similarly, inactivation of Endo U has little effect on interferon synthesis and PKR activation. I won't go, I was just going to mention, there's a completely different set of proteins in SARS-CoV-2, which we're beginning to work on. And these are the cells we're using. They have just this really pretty, these are incredible syncytia produced by SARS in the presence of ACE2 expression in A549 cells. And this is the model we're using. And I will stop there. Just to mention too, this is my group of people, some of whom working very hard in the BSL-3, and we are looking for another postdoc if anyone's interested in coming to work on. We will work on all three of these viruses, on MHV, MERS, and SARS-CoV-2. Thanks you. Sorry for going over.
No, good. Susan, thank you so much. Yeah, it's so impressive to me how these viruses work so hard to shut down the whole interferon pathway. It's obviously very important to them.
Yep.
And I would say it parallels things we only late learn about HIV, which also is very feeble in inducing any of the interferon pathway compared to somebody like Symbis, which just, you know, blows the whole system up by comparison.
But Symbis grows incredibly well despite the fact that it turns everything on. It's quite interesting.
So it has a different lifestyle, I guess, of just, you know, burn through it.
Quick. It's quick.
Yeah.
Yeah.
Okay.
All right. Did I unshare? I can't.
Yeah, you should do that.
I can't see you. That. Yeah. Huh. Oh, I'm okay. Wait. Let me. All right. Sorry.
We're good. I had one question before we move on. Just I have always wondered the extent to which the double-strand RNA exists, and are we talking very long double-strand or just a patch of double-strand behind the polymerase?
You know, we don't really know for sure. The real mystery is how does it get recognized when it's in these double vesicles where it's replicating? That's the real mystery.
Right.
And it really does seem very punctate and restricted in its localization. So that's one thing we're really puzzling about. I mean, there should be a lot of double-stranded RNA. I don't know how long it is. And these viruses are jumping. They're making transcripts and they're jumping to other regions of the genome. So it's very complicated, and we're actually trying to figure that out. And It's only detected by MDA5, which I think is kind of curious, not RIG-I.
Yeah.
Completely. It's really restricted that way.
Localization issue. I don't know.
Yep. Which is a general for all viruses have that issue.
Right. Okay. Any other takers out there?
Steve, I have a question. Sumit.
Go for it.
Susan, I enjoyed the talk. I had a question about the literature around bats and innate responses. So I've read that, you know, there's a muted innate response in bats in the reservoir. And so if that's true for MDA5 and the OAS pathway, where's the selective pressure in developing these anti-interferon activities coming from?
Okay, so first of all, that's not a universal finding that it's all muted. It's kind of, that's for particular, like there's interferon alpha is supposedly like highly expressed basally. So I think we can't really make too many blanket statements about that. In some cases it's muted, but what we saw with the OAS pathway was it was really very, very similar. Same three proteins, they're induced by interferon, they're induced by viruses, they restrict viruses. So at least for that pathway, there's really no obvious muting. So yeah, your question about the selection may be true in some systems, but I just don't think we know enough about that innate immunity to make the blanket statements.